Thanks very much, Sharon, for the opportunity to speak today. The title of my talk, it has surveillance in it, and it isn't probably truly surveillance in that I'm more going to talk about kind of more the research side of testing and talk about a little bit of an overview on the livestock diseases and what we know. And I apologize to experts on particular coronavirus diseases. We do surveillance, but it's actually done in our disease surveillance branch. Virology mainly does the epizootic testing for emergency response for avian influenza and other diseases and research. So I'm going to concentrate more on that. Most of what I'm talking about is research done with others in AFBI and others. Our vaccine research is across with Queen's University with Professor Alton Power, and I'm not going to talk about that today. Go to the first. I just want to brief overview of the bovine coronavirus involvement in bovine respiratory disease. And this is kind of one of our major research interests. And this is really a pneumonia in cattle, and it's a complex of viruses, bacteria, and mycoplasmas, but usually the viruses come in first, and we have got a research funding, a big grant to look at this as well across with the US and Ireland to look at the longitudinal, what's actually happening in the cattle. But in recent years, point out, and working with our colleagues in the US and the south of Ireland, who are actually doing testing cattle herds in the field, they are finding increasingly bovine coronavirus is coming up as one of the major viruses here. And in some cases, it is the only virus that's being found early on, certainly an infection. The virus causes cough, diarrhea, also infects the upper and lower respiratory tract, and there can be switching over with the age of the cattle. And there are a lot of questions that remain around this as to whether the respiratory infection is caused by a different variant, whether it's due to mutations, particularly in the spike protein, and also new variants, because like the viruses that they're finding in these studies are being sequenced now. This isn't published, it's kind of new work to see if the predominance is related to changes, particularly in the spike protein. So a previous study done by our colleagues in the Munster Institute of Technology in Cork actually looked at coronavirus in Irish herds, and they found that the S gene of the viruses across Ireland was different. There were mutations, and but this actually aligned with some of the European strains, closer to those. So mutations locally do seem to be occurring, and we're going to go back in a larger study looking at all viruses, which I'll mention further. So PJ Collins, you can see, is one of the people, the authors of that paper that did this work. He's now one of my senior staff, and he's very much actually focused on the porcine area now. So we will be doing a lot of work around looking at coronavirus strains in livestock. So just to mention briefly the similarities between bovine coronavirus and SARS, because I think it's quite a useful model. They're both beta coronaviruses. Spikes are quite similar. They both have the furin cleavage site. It is a different receptor in cattle. But there are, I think, other receptors for SARS as well. Also, the the actual range of symptoms in cattle can be similar to humans. And we've actually talked to our US colleagues quite a bit about this. So up to 60% of cattle are asymptomatic, and don't know what the reason for that is, whether it's genetic or down to the virus. It looks like it might be more down to the animal genetics. In the longitudinally, there's fecal shedding really in calves initially, and then nasal shedding, and the diarrhea is generally only prominent in the initial stages of infection. And there can be continual recurrent intermediate nasal shedding in some animals who are actually with or without disease symptoms. And there's only transient increases in serum antibody. So there is a suggestion that there's a lack of long-term immunity in the upper respiratory tract after natural respiratory bovine coronavirus infection. That may be similar to SARS and might be a reason why people can keep getting reinfected. And again, older cattle are much more severely affected than calves, which is obviously similar to the situation with COVID-19. Just mentioning the porcine viruses, because again, we're going to be doing, hopefully, seeking funding to do a big all-Ireland kind of trial on looking at porcine microbiomes, the viromes in pigs, and this will be done by NGS, so we will be looking at coronaviruses as well as other things. So the mentioned this morning, TGEV, transmissible enteritis, it causes diarrhea and vomiting in calves, and piglets that live more than 6 to 8 weeks after infection can recover. But they don't do well. They are stunted. But it was first reported in the States, this virus, in the 40s, and then infected Americas, Asia, and Europe. And then in the mid-1980s, TGV appeared to have mutated to a highly infectious but weakly pathogenic porcine respiratory coronavirus. And so this is a mutation which made it less pathogenic, but allowed it to spread more. But what happened then, it almost acted like a vaccine. It completely immunized or not completely, but it immunized the UK herd to TGEV, this new classified virus. So, and it's now, the incidence of both diseases in the UK and Ireland is now low and rarely diagnosed. And Julian, I think, has worked on this virus a lot, so can contradict me if any of that is not correct. But that's quite interesting because it's a virus that's mutated to be less virulent, if you like, but it's really being able to get around then more easily. So in terms of COVID, that's maybe of interest. PDV, which Sharon also mentioned, again, that was first reported in the UK in 1971 and then isolated in Belgium a few years later. Again, there are 2 genotypes there recognized, and they're split again into 1A, 1B, 2A, and 2B. But they act differently. Type 1 really affects growing pigs, whereas 2 affects all ages, including suckling pigs and mature sows. Type 1 is low-level endemic disease in the UK and Ireland, and there's proactive surveillance for that, but only detecting very low numbers annually. Type 2 is much more virulent and also known as highly virulent PEDV, and it's endemic in southern China, became endemic in 2010, and it subsequently is detected in other countries, the USA, Canada, Mexico, Austria, Belgium, France, Germany, etc. So it's now classified as notifiable in England, Scotland, and more recently in Northern Ireland. So that's PDV. Then there are a couple of other diseases in pigs, more recently recognized porcine Delta coronavirus, first reported in Hong Kong in 2012, and found in the USA, etc., and other countries. The clinical signs are less severe, and the mortality rate is 30 to 40. However, if there's co-infection with PDV or TGEV or porcine rotavirus, in fact, this leads to a more serious clinical signs if you get all the viruses together. The last then is the SARS coronavirus, the swine acute diarrhea syndrome. Again, enteric, identified first in 4 farms in Guangdong in China from 2017, so very recently, and has caused approximately 225,000 pig deaths. And gene sequencing analysis showed that this virus genome is more than 95% consistent with that of a bat coronavirus, HKU2, isolated from the Chinese chrysanthemum bat. So again, another bat-derived virus where we have that kind of information.
Louise, I'll have to ask, Louise, can I ask you to wrap up, please?
Yeah. The last slide, I was going to talk about the chicken viruses, IBD, which we can routinely test for, and turkey coronavirus. The main point just making there is that the turkey coronavirus, there is a little evidence that it also infects chickens, so species jumping, and you can infect chickens experimentally with the virus. And this, again, this is major for the poultry industry, these viruses, but they're generally tested commercially. We do BV commercially in some cases, and it's good to also distinguish from avian influenza virus. So thanks very much. Just the take-home message that the actual livestock coronaviruses, they are useful. They show us that we helpful in looking at the origins of the coronaviruses, how species jumping is occurring, that the mutations, and particularly in the spike protein, how they're changing the pathogenicity, etc. And also flag up any risk for zoonotic infection to humans. So thanks very much.